Hello and good afternoon, or good morning, or good evening, depending on where you are.
Good morning, Dr. Panesi.
I'm still working on your personal statement. When
Yes,
is
sir.
that due by? When is that due by?
December the 9th.
Okay.
December 9th.
Okay, so we have a little bit of time. Okay, good. Did you find that advice helpful?
Yes, sir. I did kind of switch it around with the points that you did give me. I think it looks a lot better now than it did before. I think the way I was writing before is kind of like the um, typical way that students would write those,
Yep,
right? So
exactly. Totally.
looking
It's
at it,
what I
yeah,
get the first time around. I'm very used to it. I hate to
yeah,
tell people, no,
yeah,
that's not the way to do it. But
yeah,
that's, I mean,
that's
that's what school's for, right?
yeah.
Hi, Shepard. How are you?
Hello, Prof. How are you?
I'm doing good. So we're going to get more into that memory of plants today that you were curious about.
All right, thank you.
What prompted you to ask that particular question? Had you come across it in some way, or I'm just curious?
I came across somewhere when I was reading.
Good. Yeah, it's did you get a chance to read those articles that I sent
Yeah,
out?
I only read two. I was left with the last one.
Okay.
I started with the last one, the second one.
Okay. Okay, good. Yeah, I'm gonna put this one in the chat just so we have it. Because I'll be talking about it a lot today if I can find it here. Let's see. Now I've put in one of the tabs on Moodle, for those of you who have access to Moodle, I've been putting a lot of these papers that I've been coming across that have been relevant to what we've been talking about. So feel free to, peruse that and read what is of interest to you. I'm uploading the PDF right now for the article on epigenetics, which we'll kind of talk a little bit about today. But I think it's usually a subject that a lot of undergraduate coursework in biology hasn't really gotten to. Maybe they mention epigenetics just a little bit. It's a relatively new field. And so I think it's appropriate to discuss more in detail in this kind of a graduate course, because it certainly plays a role in how species show their phenotypes from generation to generation. We always like to think it's just from the genes that you inherit from your parents, and that's pretty much it. We know there are environmental factors, but it's hard to imagine that there are environmental factors which affect you that will actually be passed down to your children semi-genetically, right? It's going to go with the DNA, but it's because of stuff that's happened to you in real life. And that's not usually something we think of because, the way we make our sex cells to transfer our genetic material, it's just the same genetic material you've been born with. It's just shuffled around in different ways and packaged into either sperm or eggs. And so you usually don't think that there's a mechanism by environmental factors that affect you that would actually affect your progeny like that. Unless you're talking about straight DNA damage, like due to radiation or something. So this is going to be kind of a somewhat difficult section to kind of go over because there's lots of details. I'm not looking for mastery of any specific detail, but but more so the mastery of the idea that there is this whole section of epigenetics, and that if you are looking at if your project's going to involve plants, you probably want to think about how epigenetics impacts whatever you're looking at. Because as we'll see in a minute, drought plays a huge role. You know, all of these different biotic and abiotic stressors play a big role in these things. And so if we're trying to fix our plants to deal with climate change, we're going to have to be aware of how they actually respond to some of these factors. So I'll go ahead and bring up my PowerPoint. Here we go. So this one will primarily be on epigenetics as they pertain to plants. So we talked last time about what epigenetics is, and it's defined as the study of heritable changes in gene expression. So this is talking about whether the gene's turned on or the gene's turned off. So you still have the same genes, but whether or not they actually produce messenger RNA that actually makes a protein and produces a phenotype is under this level of control of epigenetics. And so if you think of the whole person or the whole plant or the whole organism as the phenotype, it's really a combination of the DNA that they inherit and which of those genes are expressed. And the expression of those genes can be controlled or regulated by these epigenetic factors that can be heritable. So you can inherit these things and they can be passed on from one person to another, one plant to another, one animal to another. So we briefly talked about this a little bit. So it's about changes in phenotype without really changing the genotype. It's just the regulation of that gene, whether it's on or off, more active or less active. And it occurs through covalent modifications of, it can be the DNA sequence, the histones that the DNA is wrapped around. Both of those can have an impact. So DNA methylation, that's a direct way. So we're adding a methyl group onto the DNA molecule, usually at one of the bases. And that can change the activity on whether that gene can be transcribed or not. Also, histone modification. So remember, the DNA is wrapped around histones. And I can kind of show it in this little picture here. Here's what you might find in the nucleus. You know, the DNA is actually, if you unspool it, is actually loops of DNA that's looped around these nucleosomes. That's what makes up our chromatin or our chromosomes. And since it's wrapped around these nucleosomes, it has to get unwrapped in order for you to read a gene segment. So if you want to transcribe a gene that's up in this cluster here, there has to be a signal that unwinds some of the DNA loops around these histones. And this is a really important process in biology. So if you think about the differentiation of different cells, so, you know, you have heart cells, you have liver cells, you have pancreas cells. What makes those cells different? They all have the same DNA. Every single one of your cells has all of the same DNA, but a heart cell is expressing different proteins than a pancreas or a liver cell. And how is that accomplished? How is that differentiation accomplished? In part of it is by which nucleosomes get unraveled or unpacked in that particular cell, because only those genes are available to be transcribed. And there'll be different signals when cells differentiate as to which section of their chromatin they should unpack and unspool so you can get at the naked DNA. And the naked DNA is where you're going to see this transcription. And putting on a methyl group on the DNA, on the naked DNA, can have an effect on whether or not it's transcribed and made into messenger RNA. But also, there's these little tails on these histones So remember, the histones are proteins, these histone tails, that you can make modifications, covalent modifications on these tails that will either direct them to unravel or ravel back up and stay packed. And so you can limit what genes are accessed by just never unwrapping them from the histone. And that's where epigenetics really plays a role, is you can modify these histone tails to say, okay, we're never going to unwrap this region, or you can modify it and say, we're going to unravel it now so we can get access to those DNAs, or we can just play around with the naked DNA. And so it's important to kind of understand the roles of those different epigenetic modifications. Basically, your two types, that you're going to have these methyl groups, and then you're going to have these methyl groups are on the DNA, and then you're going to have these factors that are added to the histone tail. Now, there is a third type of epigenetic modification, if you will, which is non-coding RNA. Okay, so these are RNA sequences, small ones usually, that are expressed. Do you all know what antisense RNA is?
Antisense RNA. Is it, if I'm remembering correctly, is it the deletion of, not a deletion, antisense, missense, it's not deletion.
It's not deletion. Anybody else want to chime in on what antisense RNA, what that's all about? Okay, well, that's cool. If you guys don't know, that's fair enough. So remember that when you take a gene, hold on a second, see if I can bring this over. If you take a gene and you express it, you make messenger RNA, right? Well, that messenger RNA would normally go into the cytoplasm. It would make its way over to a ribosome, and then that would be translated into protein. Okay. One way to regulate certain genes, and the body does this, is to make some antisense RNA. So it's RNA that's it's like a messenger RNA, but it's complementary to the thing that you want to regulate. And so what happens if you create messenger RNA and its antisense, the two will bind together, right? Because they're complementary. And when they bind together because they're complementary, they're no longer a single strand of messenger RNA. So now you block translation. So it doesn't delete the gene, it's just silencing the signal. We call this RNA silencing or antisense RNA silencing. And so it's a way to silence the message after it's made. And so it's another level of regulation that organisms, eukaryotes, use to regulate genes. You know, you can have transcription activators, things that turn on and off transcription. You can regulate it at the level of the histones and whether or not those things unravel. You can, and for eukaryotes, you can do a lot of regulation for splicing. Antisense RNA can silence the message after it's made. It can also kind of direct splicing, because a lot of splicing occurs due to RNA molecules. So this third means is also a way of regulating the gene without changing the DNA sequence. And so you could, through epigenetics, turn on or off a non-coding region of RNA, which just codes for antisense, and you could be turning that on and that off. and thus affecting your gene. So you're not modifying your gene, you're modifying the antisense regulator of your gene. And that could be heritable on beyond the genetic code. So that's an important thing to know as well. Now, we know climate change is a big factor in what's affecting, it's affecting all organisms, and plants have to adapt to it, one, through natural selection, but their habitats are changing a lot. And before they do that natural selection, before some species just flat out die and can't reproduce, plants do try to adapt, and they try to adapt their progeny for the same environment. And so that's where this epigenetics kind of comes in. We call the things that modify the histones histone modifying complexes, and there's a number of different ones, like the compass methyltransferase complex. That's a pretty big one. There's ROS and RNS. These are key in activating oxygen and nitrogen responses in plants. They primarily affect the metabolism of the plant. And so there's a lot of these different mechanisms. There's also epigenetic mechanisms which respond to redox environments, whether you're in an oxidizing or reducing environment, and those things can hasten or slow down the modification of different histones. All of these things end up to be heritable. And I mentioned these small RNAs can also change depending on the stresses. And this little word here, sRNA, the S is for small, so this is for the small RNA genome. That's what that means. And so you have a whole section of your genome which is non-coding, which is just making these small RNAs for use as antisense regulators, like we just talked about. And so in this paper, this one here, Epigenetic Alphabet of Crop Adaptation to Climate Change, it's if you're interested or plan to do any research in plants and apply your biotechnology to plants, this is a great jumping off point to learn more about your plant and how it responds on an epigenetic level. Because you might want to look at, okay, can we change the drought tolerance of this plant? First, you want to understand, how does the plant respond to drought innately? And, you might know the genome of the plant, but do you know its epigenetic state? And does modifying that epigenetic state help it adapt to the climate change environment? And so here's one of the figures from that paper. And what they're trying to do is come up with, we have an alphabet for the genetic code, A, G, C, and T, right? And so now they want to add on to this alphabet these different histone and DNA modifications. They don't really talk about RNA modifications in this paper. It's primarily the histone and the DNA modifications. And you can see that they've divided the types of stresses that lead to epigenetic modification into a couple different classes. Drought, hyperosmotic, different levels of salinity, whether it's hot or whether it's cold, light, the amount of light that it's getting, that also affects the epigenetics, as well as the metals that are present. And what they've listed here are several, on this bottom panel here, are several of the biological mechanisms that we know of that do epigenetic modifications, a set of enzymes that'll either put an acetyl group on a histone or a methyl group on DNA. These are the names of some of those complexes that we know about. There are probably many more that we don't know about, but we're slowly deciphering this. And so, this becomes a quick guide. If you're trying to look at drought tolerance in plants, you look for these checked boxes, which are the different groups of enzymes or complexes that are involved in, say, drought tolerance. And these would be some of the ones that you would want to measure in your particular plant if you were looking at adjusting its drought tolerance, right? Because you want to know how these things affect stuff. So here's a picture also from the text, just talking about the role of some of these different things. A lot of the HDAs and the HATs, those are all mechanisms or complexes that are geared around acetylation of the nucleosomes, so they unfurl or stay together. The DMTs and the HMTs, those are the methyl transferases, and so those will directly affect the DNA generally. So it's primarily acetylation and methylation. Ubiquitin is also added on in some cases to these nucleosomes as well. And so you've got a whole different set of classes that kind of tell you, which systems you're going to favor. If you're in drought, looking at the drought genes, a lot of these are methyltransferases that are involved in that, and there are just a few that are involved in acetylation of the histones and histone modification. And so they break the paper into looking at a bunch of different stressors. So one of them is drought. Okay. And this is something that we really expect to see a lot with climate change. We know that dry areas are going to get drier and the wet areas are going to get wetter. And we know that more of the precipitation is going to come in larger events, and the events will become increasingly further apart. So even if you're in a wet region, you could still experience droughts, but then you get deluges of water in between your drought, which isn't great because when the ground is really dry, it has a hard time absorbing a lot of water really fast. And so if you have a huge deluge, rather than just absorbing all that water into the ground, you'll probably end up with a little absorption and a lot of flooding. And then you'll go back to drought for a long period of time. And so this can lead to water scarcity, even in places that are predicted to be wetter. Because remember, as you have warmer air, it's going to absorb more moisture, and it can tolerate more moisture before it rains. So the areas around it will get drier. So here's just a couple examples for droughts. For example, they know in this particular species here, this complex here oversees histone acetylation in response to drought and affects some of the nitrogen metabolism and the gene expression there. And these kind of form like what we call a stress memory for the plants, which they pass on, letting the new plants that grow know that they're already in a stressed or drought-ridden area. And so certain genes will be upregulated. Again, DNA methylation is a really big role that's used in drought. It can be reversible or irreversible. The irreversible ones tend to produce all progeny from those plants tend to not be able to handle when the drought goes away, for example. These changes will persist through several generations, affecting the offspring stress response, etc. So it's something to be really mindful of. Okay, there is some research, and the paper refers to this on some of the microRNAs or the small RNAs, antisense RNA regulators that also play a role in drought. And one of the reasons why I like to show this paper and discuss this paper isn't so much for any particular factor that's in it, but for, if you look at the bottom of the paper, go all the way to the back, you get to the bottom here, see all these references. This is really the gold mine of this paper. So it's not that you need to understand every single kind of stressor or what the response regulator is for that stressor. It's that if you're working in a particular species and you're looking in that particular stressor, it will have a reference on, okay, read this paper, and you'll be able to find out, more about, so like this is looking at woody plants, for example. So in this set of references is a gold mine of other papers that you definitely need to read if you were working on a project in any particular plant. You wouldn't read all of these, you just find the ones that kind of apply to what your particular project is. So in addition to drought, they have several paragraphs talking about salinity. Okay. This can cause hyperosmotic stress and can inhibit plant development. So salinity is about the amount of salt that's present, level of salt that's present in the water. And again, this can create methylations that are passed on to progeny, so you still get the stress memory. And it's been seen in soybean and rice. They have demethylation at a sodium transporter pump, which increases the salt tolerance. In other cases, you'll see methylation improving tolerance as well. So it can be demethylation or methylation, just depending on the specific thing that you're looking for. There's a lot of research on soybean and rice and a lot of references to that. So if that was a project you were interested in, you'd probably get those papers. Specifically in rice, we've seen that a lot of the zinc finger genes have are really important for DNA regulation. And when we're in the next course in genetic engineering 2, when we cover protein structure and function, we're going to go over what zinc finger genes are and what zinc fingers are in proteins and what they do and all of that. So try and remember that those zinc finger genes do play a role in how plants respond to salinity as a stress. Also, I know in Africa, millet is a big product that people rely on. And so if that's a project you're interested in, there are several papers that talk about what we know about epigenetics in millet crops as it relates to salinity and such. There's also articles about wheat and corn. and how they respond to salinity as well as beets and alfalfa. And so what's neat about the research now is it's become very species specific, especially for a lot of the, crop type plants, where we're starting to learn more about the epigenetics that kind of control how they respond to these different stresses. So when you get into Dr. Chowdhury's, I think it's not next semester, it's the semester after that, you'll be taking plant biotechnology. He's going to go over a lot of this stuff with you. And so this is just kind of a primer to let you know epigenetics is important. But if there's a particular crop or a particular plant that you're interested in as part of your capstone project, or just something that you're interested in, let me know, and I can kind of give him a heads up as well. But you'd want to start reading some of the papers related to that are mentioned in this paper, in this review article. In addition to salinity, obviously temperature is a huge role, a key factor affecting plant growth, right? And both high temperatures and low temperatures have limiting effects on how plants grow, okay? And there's many examples of epigenetic processes for many different species that just deal with temperature in general. So they've looked at cold stress in rice and found about 29 different genes that are affected through DNA methylation in rice when it's cold out. And usually these are methylations occurring at the promoter regions. They've seen histone modifications in gene silencing as well in heat and cold stress responses. And that affects chromatin accessibility, how unraveled these things become. There's even light triggers that affect epigenetics. So if you think about if a plant is trying to deal with changes in the amount of shade that it gets, like one area is shaded and one's not, we know that the plant grows towards the light. Why does it only grow towards the light and not just evenly? It's actually a large factor in controlling that are these histone modifications, okay, as well as methylation. So these epigenetics turn on and off certain genes that allow it to grow in particular directions. Also, the amount of UV light has an effect on DNA methylation in plants, and that can be transcribed as well. Let's see. And even heavy metals can have a huge effect on the epigenetics of plants. So we know from what we've learned about climate change that as you increase the amount of CO2 in the atmosphere, more of that CO2 is going to get absorbed into the rain. And so that creates that carbonic acid, which when it rains down, we know leads to chemical weathering. And that's one of the ways that carbon gets into the rock reservoir, right? We talked about that. Well, in that chemical process of getting the carbon into the rock reservoir, sometimes releases heavy metals because there's an exchange. You're putting carbon in instead of some other element. And so certain elements can be released. Often these are heavy metals. And now that's in the environment, and that can be around for plants. If you have lots of precipitation, you're going to get lots of runoff that runoff is going to make its way into the oceans. So you're going to have more heavy metals into the seas, etc. So one example of that are soil arsenic levels have been shown to rise because of weathering. And that leads to a direct DNA methylation in Arundodonax, which is this plant over here. You've probably seen before. It's fairly common, and it's basically a tall grass that's very common. But you see it already responding to arsenic levels in the soil that are a response because of climate change. Another element that's released because of climate change is aluminum. And that can affect rice, wheat, and tobacco, or at least it can probably affect more. But when I mention wheat and tobacco, that's because there's several papers on those specific plants looking at the effects of aluminum in the soil and how it's affecting the growth of those plants. Take a break here because this is like a lot of information. So what are your thoughts? First off, did you know about epigenetics before? And what do you think about them now?
I knew kind of a little bit of the concept of epigenetics, but not so much in detail as I do now.
Okay. Shepard, does this answer some of the questions you had about memory in plants and how that works?
Hello, Prof. Yeah, I think I'm getting more detailed into even I can come up with more like a proposal in the area.
Okay. Okay. Glory, how about you? Are you following along with this so far?
Yes, Prof. Actually, I was never aware of epigenetics. It's quite interesting, and I'm glad that I'm learning this.
Good,
thing is
good. Brenda, Zakiu, Naimungo, do you have anything to add or any questions so far?
Personally,
No,
I'm
sir.
just following.
Okay.
Maybe when we finish, I'll be able to ask questions.
For me,
Okay.
everything's okay. I got everything. Now I know more about epigenetics.
Okay. So for each of these, each one of these things I mentioned, I could probably do a whole lecture on exactly what is the signal that comes in for the environment, how does that signal make it into the plant, then what particular system does it activate? Like if it's going to be a histone acetylase that's going to acetylate a histone and cause a particular region to wind back up and not be accessible for transcription. We could spend a whole lecture on just one of those. And as you've seen, there's hundreds of them, right? And so that's not really the point of this course in Introduction to Climate Change, is to go through each and every one of them or show you tons of them. But I want you to know that they're there, because when if you'll learn a lot more about plant biotechnology and plant genetics and how to grow them and how to change them and how to play around with their genetics, in further courses. But I think it's helpful to know what you would need to research so you can be kind of aware of these effects, because there are definitely real effects. A lot of farmers that don't know about this are trying to replant plants and they're not growing like they used to. And they're like, well, what is it? Well, a lot of times it's the memory of the plant itself. And, it's like the plant has PTSD. It's not doing too well. And so it's kind of important to know about. So rather than focus whole class periods on different systems, I just wanted to show you the breadth of these different systems and then give you this guide paper, this epigenetic alphabet paper that can kind of guide you where to look and how to look if you want to pursue any particular plant further. Because in the end, really the knowledge is only important when you want to apply it, you know, when you're working on a specific problem. And you may end up working on problems that have nothing to do with plants, and that's fine, but at least know that epigenetics are out there. The other thing I wanted to mention is don't forget that there is a peer teaching that is due that y'all should be working on. I have one from Caldrian, but nobody else yet. So I'm gonna, in the chat, I'm gonna put the link to the prompt. And remember, in this one, you're gonna be describing an effect of climate change or something that's exacerbated by climate change in detail. It can be something that we've already talked about in the class. I prefer they to be something that's particular to your geographic area or something that you would be interested in working on. If you've got an idea for what your capstone project might be in a year and a half, and you want to start thinking about it now, this is a great place to introduce it. And I can't wait to share these with everybody because I think you're all going to find each other's videos fascinating. I've talked to a couple of you about what kind of projects you might be interested in and climate change effects that you're fairly passionate about. So I'd like to see some in-depth dives. Remember, they're only 5-minute videos, but I'd like to see slides and as much data as you can put in there, related as much as you can to the concepts in the course, quantitate things where possible, to have this a nice little intro into the topic that you might pursue further on. Now, you're not bound by this, so if you do something, you know, for this peer teaching now, in a few months, if you've come up with a different idea for a capstone project, that's totally fine. You're not bound by this. But I do want to see something that you've taken out of the course or something that you've thought about in terms of impact of climate change. Are there any other topics? So we've got one more lecture this week on Friday. Is there any other effect of climate change that anybody would like me to talk about that we haven't covered so far? Because once we're done with those effects, I mean, I could do lecture upon lecture upon lecture of different effects. But what I want to get to in the last part of the course is some of the policy initiatives and how people are trying to fight climate change. Like, what are the Paris Accords? You know, what are some of the things that the IPCC has agreed upon or that countries are negotiating to try and change which of those climate scenarios we find ourselves in. But is there any climate change effect that anybody would like me to go over?
I was going to add maybe ways in which countries are coming together to fight a particular effect of climate change.
Oh, okay. Okay. One case study, if you will. Okay, great. Any particular effect of climate change you're most interested in?
I will use I would use a cliche of course warming temperatures, but that's the only thing that comes to mind. Maybe drought.
Okay.
In the country of Africa, or extreme cold, kind of in Russia. Maybe either or, but
Okay. Any other topics?
Hello, Prof. I want to ask you, are we also allowed to do like a qualitative paper where you are focusing on let's say adaptation ways farmers are using to adapt, let's say, for climate change? then you'll be asking them in form of survey. Then you end up collecting data, let's say for a sample size of around 300 farmers and above. Then you document and you come up with a paper.
Oh, that's a fabulous, that's something fabulous. I would love to see. It wouldn't fit into our 5-minute video. So I would try and, maybe summarize or introduce that topic, exactly, how they're going about. But put together something you can in a 5-minute video. I would like to talk to you further about what folks are doing. One particular topic I kind of am interested in, and have you all heard of the fall armyworm? So Dr. McCutcheon here, actually, she's an entomologist. It used to be a fairly problematic species in the Americas, in North and South America. It's recently, I think around 1996, jumped to Africa and become a horribly invasive species that's affecting corn, mullet, all sorts of stuff. And it's really been going rampant. Got some neat articles on it. And actually, as part of her, I think it may have been her thesis, she had done some research on how to combat it using native predators and introducing native predator species. And we've actually come up with a possible project if somebody in Africa wanted to do this project as part of their capstone project to fight fall armyworm. That might be very doable. We've got a lot of data on it, and we're poised to do a project. But that was one of the projects we mentioned at the Africando conference when we were talking to folks from a variety of different countries in Africa, that they seemed to be interested in, that we would have students on the ground there working on fall armyworm and combating it. So in your surveys, when you're talking to folks, farmers, ask about the fall armyworm, because I'm really interested in hearing about that as well. Thanks, everybody. Any other questions? Comments? You guys happy with the class so far? Good. Oh,
Yeah.
okay.
Yes, sir. Yeah.
Okay. I'll be looking for your responses soon, and I'll see you on Friday. Thanks a lot.
Thank you, Prof.
Bye-bye.
Thank you.